everyone, it's Dr. Mohib, and in this lecture we'll be going through amino acids, ammonia disposal, hyperammonemia, urea cycle and its disorders like ornithine transcarbamylase deficiency, and at last some memory drills. So first of all, amino acids. Amino acid is the building block of proteins. The foods we eat generally lies in one of the categories of macronutrients like carbs, proteins, and fats. The building block or the monomer of proteins are amino acids. Protein exists in such a way that they consist of large peptides, which are the polymers. They are then broken down into smaller peptides or di or tripeptides, and they are further broken down into individual amino acids. So this was about amino acid. Now what is amino acid? Amino acid is a carbon structure consisting of two functional groups, a carboxylic group at one end and amino group at another end, and an R side chain. This is the structure of amino acid, and each amino group differ from another by this R side chain, and the simplest amino acid is glycine, in which there is a simple hydrogen instead of the R side chain. Now the amino acids can exist in two configuration, in L configuration as well as L configuration, also known as LEVO configuration or left sided configuration, and D configuration or dextro configuration. In L configuration or LEVO configuration, the amino group lies on the left side of the amino acid, while in dextro configuration this amino group would be on the right side. Surprisingly, our body can only metabolize the L form of amino acids. So only L amino acids are found in protein that are metabolized by our body. The amino acids are divided into two categories: essential and non-essential. Essential are those amino acids which our body cannot synthesize, and they must be taken up in diet, while non-essential amino acids are those amino acids which our body can synthesize depending on the need of the body. So essential amino acids are remembered by the mnemonic private Tim Hall or PVT Tim Hall. The PVT: phenylalanine, valine and tryptophan. Then Tim Hall. Then the Tim stands for threonine, isoleucine and methionine, and Hall: histidine, leucine and lysine. So these are all essential amino acids. Now there are two categories, or rather three categories of amino acids depending on the products of metabolism. They can be either glucogenic, which yields pyruvate, or TCA cycle intermediate, while ketogenic are those which yield acetyl CoA or ketone bodies, while glucogenic and ketogenic are the mixed type of amino acids which yields both pyruvate or TCA cycle intermediates or acetyl CoA or ketones. So I have discussed it in the last lectures or the TCA cycle lectures, but for brief review over here, the pyruvate or TCA cycle intermediates can be converted into glucose, because glucose oxaloacetate is converted into phosphoenol pyruvate, which goes on to develop into glucose via gluconeogenesis, and pyruvate can also be converted into oxaloacetate. Pyruvate is converted into oxaloacetate. So either pyruvate or any of the TCA cycle intermediate can be converted into glucose via gluconeogenesis. Thus, those amino acids are known as glucogenic amino acids. If you look at over here, like the vomit mnemonic or the valine, isoleucine, methionine, threonine, they are converted into propionyl CoA, which goes on to develop into methylmalonyl CoA, and succinyl CoA, which goes on to develop into oxaloacetate and phosphoenol pyruvate, and at last into glucose. So this was all about glucogenic amino acids. Then we have ketogenic. Ketogenic are converted into acetyl CoA or ketone bodies. To remember, the ketogenic are easiest to remember. The mnemonic is the only purely ketogenic amino acid, or those starting with L or the last two. So the leucine and lysine both are purely ketogenic amino acids, while the glucogenic and ketogenic mix are remembered by the mnemonic PIT. So phenylalanine, isoleucine, threonine and tryptophan. the phenylalanine, isoleucine, threonine, and tryptophan. They are the mix, while the lemanic the are glucogenic, like methionine, histidine, and valine. The mnemonic is we met his Valentine who is so sweet. Sweet is for glucogenic. So M for methionine, histidine, and valentine. So this was all about the essential, non-essential, glucogenic, and ketogenic amino acids. Then we have another type of amino acids like standard amino acids and non-standard amino acids. The standard amino acids are those amino acids which are encoded by our genome, while non-standard are those amino acids which are not encoded by our genome. They consist of amino acids, carboxylic acids, the keto group, but they are not encoded by our genome, but rather they are the products of either urea cycle or other cycle. Then the other thing to remember about amino acids are they can be either acidic or basic. Now to understand it, if we consider a substance or an acid which can exist in two states, either in H++, A-, either in associated, these are the two states. So if we write the dissociation constant for it, log dissociation constant, it would look like somewhat like this. So if a substance or a reaction in which the concentration of both A minus, the dissociated form, or the undissociated form are equal, so they will cancel out each other, and the log dissociation curve or the pK of the reaction or the substance would be equal to the pH of the solution. Now, if the pH of any solution is greater than the pKa of that substance, for instance, if the pH is greater than the pKa of that substance, this means that the hydrogen ions in that solution will be very low, and the substance might lose electrons and would exist in the negative form. So if a substance is like this, it will lose electron and will exist in the negative state. So if the pKa is lower than the pH of the solution, which is in the case of acidic amino acids. The acidic amino acids have low pKa. So in the body pH, which is 7.4, in this state, the acidic amino acids exist in the negative state. So they are the two acidic amino acids, as their name implies. They are aspartic acid and glutamic acid, also known as aspartate and glutamate. They exist in the negatively charged state at body pH, while for basic it is the opposite of it. So in basic, the pKa of amino acid is actually greater than the pH of our body, which is 7.4. This means that we will have a lot of H plus. This means that the solution or the amino acid will exist in the positive state. So the amino acids or the basic amino acids exist in the positive, or they have the positive charge. The examples are histidine, lysine, and arginine. Remembered by the mnemonic, his lies are basic. Those having girlfriend and boyfriend relationship would refer to this mnemonic. So his lies are basic: histidine, lysine, and arginine. While arginine is the most basic. Histidine has no charge at body pH, although it is basic, but it has no charge. Arginine and histidine having the positive charge are required during periods of growth, because these are required during periods of growth, and I have confused it or mixed it. The arginine and histidine are required during periods of growth, while arginine and lysine are in histones which bind the negatively charged DNA because they carry the positive charge. So this was all about amino acids. Now let's come toward ammonia. Unlike the carbohydrates and fats, the other macros which can be stored in our body, there is no storage form for amino acids, so they must be metabolized. So amino acid catabolism generates common metabolites like pyruvate, which is a product of glucogenic amino acids, while acetyl CoA, which is a product of ketogenic amino acids. And during the catabolism, or those amino acids which are not required in our body, they must be excreted because they yield ammonia, and Ammonia is increased ammonia is toxic to our body. So how does amino acid metabolism occur? So we have amino acid. So when amino acid loses amino group or ammonia, it is converted into alpha keto acid. The examples are when an amino acid glutamate loses amino group, it is converted into alpha ketoglutarate. Same goes to aspartate. When aspartate loses amino group, it is converted into oxaloacetate, which is the keto acid of the aspartate. Then we have alanine, and when alanine loses amino group, it is converted into pyruvate. So this is the metabolism of amino acids. Now what happens in our body? So whenever the body has an excess of amino acids, they are metabolized. So for instance, we have an amino acid, it is converted into alpha keto acid, and the amino group which is lost, or the amino acid which loses its amino group, it is given to alpha ketoglutarate, and alpha ketoglutarate is converted into glutamate, and this glutamate now carries the amino group of the amino acids being metabolized. Now this glutamate must go to liver, where this amino group is converted into a non-toxic substance like urea. This amino group must go to the liver, where it is converted into non-toxic substance urea, which is excreted renally. Now this glutamate can go to liver by two methods or two ways. The first is it can go in the form of glutamine, and the second way is by alanine, which is also known as Cahill cycle, and this method is used by the muscles. So first of all, glutamine. So whenever an amino acid is converted into its alpha keto acid and amino group is released, this amino group is taken up by alpha ketoglutarate, converting into glutamate by an enzyme glutamate dehydrogenase. Then the glutamate is further converted into glutamine, adding another amino group by an enzyme glutamine synthase. Now this glutamine can go to, this exists in tissues, while this glutamine can then go on to liver, where the exact opposite occurs. Glutamine is converted into glutamate, and glutamate is converted into alpha ketoglutarate. And when glutamate is converted into, glutamine is converted into glutamate by an enzyme glutaminase, and by doing so an amino group is released. Same when glutamate is converted into alpha ketoglutarate by glutamate dehydrogenase, and amino group is released. These amino groups can then enter the urea cycle, where they are converted into urea. The second method of transfer of glutamate's amino group is by alanine. So what happens is, this is the muscle. In muscle, amino acid is converted into its respective alpha keto acid, and amino group is given to the alpha ketoglutarate, converting it into glutamate. Then glutamate gives the amino group to pyruvate, converting it into alanine. This alanine then enters the bloodstream, enters the liver. In liver, the opposite occurs. Alanine gives its amino group to alpha ketoglutarate, converting it into glutamate, which can give its amino group to enter urea cycle, while alanine is converted into pyruvate. And these reactions are carried out by amino transferases. All these transfers of amino group reactions are carried out by amino transferases, which require B6 as a cofactor. In this case, it is alanine transferase, which is usually elevated in liver pathologies. This pyruvate then is converted into glucose via gluconeogenesis. The pyruvate then again enters the bloodstream, goes to glucose, goes to muscle, where it is converted into pyruvate by glycolysis, and the process again continues. So this was all about transfer of glutamate's amino group by alanine or Cahill cycle and glutamine. Now, if there is any pathology or any sort of disturbance in handling of this ammonia, and the ammonia increases in our body, which leads to a state known as hyperammonemia. It can be due to two reasons. Either there is a there is some sort of pathology in liver, where liver cannot handle this ammonia, or there is some sort of pathology in urea cycle which handles this aminos or amino groups or ammonia. And why does this high ammonia or high NH3 is detrimental for our body? The reason is that it is CNS toxic. Increased amino levels is toxic to our CNS. The reason they are toxic is that the number one is that when there are a lot of ammonia in our body, it is detoxified by alpha ketoglutarate, converting it into glutamate, and in doing so the alpha ketoglutarate levels drops, and we all know that alpha ketoglutarate is a TCA cycle intermediate. So this means that the TCA cycle will halt or stop. The second reason is that if there are a lot of ammonia, the glutamate is further converted into glutamine, and the glutamate levels are decreased, and we all know that glutamate is the stimulatory neurochemical substance. Then the conditions or these chemical states of our body leads to the conversion of glutamate to GABA, or in other words it increases the inhibitory tone, as the GABA is the inhibitory neurotransmitter of our brain. So increased GABAergic tone, or GABA is increased. We are, as we have mentioned, that there is the glutamate is converted into glutamine, which leads to increased glutamine, and these glutamines are taken up by the astrocytes of brain, and this glutamine is actually an osmotic substance, meaning that it will pull water also with it, thus causing cerebral edema. Glutamine induced osmotic shifts. Thus, due to all of these, the patient will have the following symptom. The classic symptom will be flapping tremors, or also known as asterixis. If we ask the patient to stretch out their hands will start to tremble. This is known as asterixis. The patient may also have slurring of speech, somnolence, vomiting, cerebral edema, blurring of vision, memory impairment, all of the CNS symptoms due to the high ammonia levels. Now, how can we treat this? As we know that these aminos, they come from proteins metabolism. So if we limit the protein in diet, we can somewhat control the levels of aminos in our body. The second method is to give some certain drugs which can decrease the ammonia levels. The very first drug is lactulose, which is laxative to acidify GI tract. The lactulose is metabolized to fatty acids, which decrease the pH of the GI tract, and thus due to decreased pH, the ammonia is converted into amino group, or since the pH is less than the pK, the high hydrogen ions will convert ammonia into NH4 plus. The NH4 is impermeable, or the NH4 cannot be absorbed. Thus, the ammonia in our diet cannot be absorbed and excreted. The second method is by giving certain antibiotics like rifaximin to decrease aminogenic bacteria. There are certain bacteria in our GI tract which synthesize ammonia. So by giving antibiotics like rifaximin, we can kill those bacteria. Then the third option is giving certain drugs like benzoate, phenylacetate, or phenylbutyrate. They react with glycine or glutamine, forming products that are excreted renally, and these drugs are reserved for children having urea cycle disorders. So this was all about hyperammonemia. Now let's come toward urea cycle. Now what happens in urea cycle is that excess nitrogen is converted to urea, and this urea is excreted by the kidneys. This urea consists or carries two amino groups, or its structure consists of a keto group and attached to it are two amino groups. One amino group comes from ammonia, the keto group or the carbon skeleton is from carbon dioxide, and the last amino group comes from the amino acid aspartate. The reaction is as follows. The amino group combines with the carbon dioxide. In certain books it is written carbon dioxide, while in certain books carbon dioxide combined with water, it is converted into bicarbonate, and bicarbonate and ammonia combine to form a structure known as carbamyl phosphate by an enzyme carbamyl phosphate synthetase 1 using 2 ATP and N-acetyl glutamate, which is an allosteric activator of this reaction. So we have a product carbamyl phosphate, and all of these reactions take place in mitochondria. One thing to remember about urea cycle is that it takes place in both mitochondria and cytoplasm. The mnemonic for it was that hugs take to the heme synthesis, urea cycle, and gluconeogenesis all took place both in mitochondria as well as cytoplasm. Now, this carbamyl phosphate is also a product of another metabolism, which is nucleotide metabolism. In nucleotide metabolism, for the pyrimidine, the base pyrimidine metabolism, glutamine combined with carbon dioxide to form carbamyl phosphate by an enzyme carbamyl phosphate synthetase 2. So it is an intermediate both in nucleotide metabolism as well as urea cycle. This carbamyl phosphate combined with ornithine to form citrulline. This reaction is carried out by an enzyme ornithine transcarbamylase. Then citrulline converted into arginosuccinate using an enzyme arginosuccinate synthetase, and aspartate is added and ATP is used in this reaction. This aspartate gives the another amino group to the urea cycle. Then arginosuccinate is converted into arginine and fumarate is released, and the enzyme carrying out this reaction is arginosuccinate lyase. This fumarate can then go on and enter the TCA cycle over here. If you look at over here, arginosuccinate gives arginine and fumarate, which is a TCA cycle intermediate, which can enter the TCA cycle. So it is the crossroad or link between urea cycle and TCA cycle. Then arginine is converted back into ornithine, and using an enzyme arginase, urea is released which goes to toward kidney for excretion. The first two reactions, conversion of the carbamyl phosphate combining with ornithine to form citrulline, and then this citrulline leaves mitochondria. The rest of the reactions takes place in the cytoplasm, and the ornithine formed over here enter again mitochondria. The mnemonic for it is, I have personally used this mnemonic, that the coca are from AU, Australian Union, or you can learn however it is convenient for you. C for carbamyl phosphate, and ornithine combine to form citrulline. Then after that we have aspartate combining to form arginosuccinate. Then arginosuccinate fumarate is released, giving off arginine, and at last urea is given. So I personally use this mnemonic, while if this mnemonic is convenient for you, you can use this ordinarily. Careless scrapers are also frivolous about urination. So this was all about urea cycle. Now, if there is any sort of disturbance in, or an enzyme is deficient in urea cycle, for instance, if we have an ornithine transcarbamylase deficiency, so we will have increased ammonia and signs of hyperammonemia, because the ammonia will not be handled properly or converted into urea, and other than that, the reactants or the products backward of the enzyme will increase also. So in case of ornithine transcarbamylase, we will have high levels of carbamyl phosphate. In case of arginosuccinate synthetase, we will have high levels of citrulline in addition to hyperammonemia. In case of arginosuccinate lyase, we will have high levels of arginosuccinate, and in case of arginase deficiency, we will have high levels of arginine. The rate limiting step of urea cycle is carbamyl phosphate synthetase 1 reaction, or the conversion of ammonia into carbamyl phosphate, and N-acetylglutamate is an allosteric activator of this reaction. This N-acetylglutamate is synthesized from glutamate and plus acetyl CoA, meaning that they can be synthesized by having high protein meal. So after taking high protein meal, we will have high levels of N-acetylglutamate, which is the allosteric activator of carbamyl phosphate synthetase. So this was all about urea cycle. Now let's review the famous or the most common urea cycle disorder, which is ornithine transcarbamylase deficiency. So in ornithine transcarbamylase deficiency, it is the most common urea cycle disorder, as well as it is the only X-linked recessive disorder, meaning that it will affect only males. All other urea cycle enzyme deficiencies are autosomal recessive. It interferes with the body's ability to eliminate ammonia, as urea cycle is the process of eliminating ammonia from our body. So it is evident, often evident in the first few days of life, but may present later. So the patient will have CNS symptoms like the kid will be somnolent or irritable, etc. The excess carbamyl phosphate, as I have mentioned earlier, that in ornithine transcarbamylase deficiency, we will have high levels of carbamyl phosphate. These high levels of carbamyl phosphate is converted into orotic acid, which is the part of the pyrimidine synthesis pathway, versus carbamyl phosphate synthetase 1 deficiency. So if I refer again, so the carbamyl phosphate is the common product of both pyrimidine metabolism or synthesis pathway, as well as urea cycle. So this excess carbamyl phosphate will be converted into orotic acid. So we will have high levels of orotic acid in blood and urine. We will have low levels of urea because urea cycle is disrupted, and symptoms of hyperammonemia. No megaloblastic anemia versus orotic aciduria. Now, ornithine transcarbamylase is often confused with another disorder, which is the disorder of pyrimidine synthesis or pyrimidine metabolism, in which there is an absent, this orotic acid combines with the phosphoribosyl pyrophosphate to form UMP, uridine monophosphate, and the enzymes carrying out this reaction is UMP synthetase, which is absent or impaired in orotic aciduria. So in orotic aciduria, we will also have high levels of orotic acid. But unlike ornithine transcarbamylase deficiency, the patient will not have symptoms of high levels of ammonia. And other than that, the orotic acid patients will have megaloblastic anemia, because it goes on to develop into uridine and cytidine. So these patients also have megaloblastic anemia. So this was all about urea cycle and its enzyme deficiencies. Now let's review. Now let's have some memory drills. OTC deficiency is inherited as autosomal dominant, autosomal recessive, X-linked recessive, or mitochondrial. And the correct option is X-linked recessive. The second is cerebral edema in hyperammonemia is mainly due to lactate accumulation, glutamine-induced osmotic shifts, increased ATP, or hyperglycemia. And the correct option is glutamine-induced osmotic shifts. And the last is a patient with hyperammonemia and elevated orotic acid most likely has high levels of ammonia and high levels of orotic acid. So is it OTC deficiency, orotic transcarbamylase deficiency, phenylketonuria, alkaptonuria, or maple syrup urine disease? And the correct option is OTC deficiency. So this was all about amino acids. Thank you.